Hi everyone, and welcome back to Biology 206. My name is Dr. Jennifer Baltzer, and we're going to be continuing our conversation about populations today, focusing on distribution and abundance. And we're going to start with this mystery of the kelp forests. Like trees in a forest, kelp provide the structure of these offshore, these coastal environments off of the west coast of North America. These huge, here we have bull kelp or laminaria, which grow to great heights and support all kinds of biologic diversity and ecosystem productivity in these environments. But the distribution of these kelp forests has been declining, and this for some time was a bit of a mystery to researchers. There were areas where kelp were being maintained and other areas where the kelp forests were declining, and some of the initial investigations really focused on physical drivers. Are there physical differences in the places where kelp forests were remaining intact and where they were disappearing? And that appeared not to be the case. And so researchers started to investigate some additional factors that have to do with biotic interactions in these communities. And an important factor are these purple urchins, sea urchins. These sea urchins basically move across these kelp forests and kind of raise the kelp forest. So they move across the surface, feeding and along the base, along the floor where the kelp are rooted, and this leads to basically a clear cut of this kelp forest. And researchers demonstrated the important role of these urchins in a couple of ways. One was observational studies, so where they went out and they looked at the relationship between urchin abundance and the condition of the kelp forest, and found where urchin abundance was high, that the condition of the kelp forest was very poor. And then secondly, they did experimental studies where they actually actively removed sea urchins and looked at the density of laminaria. So using area-based sampling, where they remove the urchins in some places and leave the urchins in other places, and found that where they were experimentally removed urchins, recovery was fairly rapid. So before urchin removal, there was no kelp. 1 year after urchin removal, kelp density was at 20 individuals per meter squared, and 2 years after removal, kelp density was up to over 100 individuals per meter squared. So this is what we have on this axis, is from 0 to 110 individuals per meter squared. Okay, so clearly urchins are playing a really important role. But then the question was, well, why are there these differences in urchin densities? What's happening between these different regions that's driving differences in how many urchins there are and their role in these ecosystems? And we will continue this toward the end of the lecture. We'll solve this mystery in a little bit. Okay, so today's lecture, now that we moved on from the introduction of our mystery, today's lecture is really focused on distribution and abundance. And we can see this demonstrated in these tropical woody species here. So Brosimum alocastrum, Cockaloba coronata, and Chamguava shippii. And each of these dots represents an individual in those populations. And you can see these very, very different distributions that correspond to what we see in these circles as well. So random distribution, where we've got individuals distributed across the landscape in sort of a haphazard fashion. Uniform distribution, where we have pretty consistent, even spacing of individuals, and then a clumped distribution where individuals of this species are only located in one particular place. And so you can see this demonstrated here in these petri dishes as well, where we have our random distribution, a very even or uniform distribution, and then this clustered or clumped distribution. And we'll get into that in a little bit. But what I want to talk about first is movement between populations. And so we have a couple of really important terms that are going to be terms that we spend a bit of time on during this lecture. The first being dispersal, or the movement of individuals into and out of an existing population. So the movement of individuals into a population is immigration. The movement of individuals out of a population is emigration. And we can think about this in the context of what we refer to as the BIDE factors. So the population size is going to be a function of births and deaths, so B and D, as well as immigration and emigration. So births, immigration, deaths, emigration. So births and immigration are individuals coming into a population, deaths and emigration are individuals going out of a population. So those immigration and emigration factors are a function of dispersal, or the movement of individuals from one population to another. And this links really closely with metapopulation. So when we think about populations, a lot of times they are not continuous. And in fact, you have something akin to what we see here in this distribution of Clematis fremontii, which we see here, this lovely little flower, bell-shaped flower. So this Clematis species, we have its distribution here. So it has a range, a geographic range where it's distributed in this little corner of Missouri. Within that little corner of Missouri, there is what we refer to as a metapopulation. All of these different red areas where that species is found. And so each of those red areas is a population. It's isolated from all of the other populations within the metapopulation. But because of dispersal, there's movement of individuals between one patch and another, okay? And I apologize if you hear my dog crying in the background. Okay, so back to this idea that I introduced a little bit earlier. We have different distributions within populations, and this can be driven by different processes. So again, we've already talked about this a little bit, but this is from your text: a regular or evenly spaced distribution, a random distribution where you have random or really haphazard spacing, and a clumped distribution where individuals are grouped together. And this can be driven by different factors. In the case of, a random distribution suggests that there's not a lot of control over where those individuals are recruiting into the population. A regular distribution suggests that there's some factors that require individuals to be spaced a certain distance apart. And a clump distribution suggests that there's some, attracting factors that lead to individuals all occurring in the same environment. A great example of this sort of clumped distribution comes from these lovely towering forests of Southeast Asia, the Dipterocarp forests. Here are the amazing seeds of different taxa in the Dipterocarpaceae. And here we have the distribution of 5 different species of Dipterocarps in a rainforest, in a mapped forest plot in Sarawak on the island of Borneo. So this is Lambir Hills, and there's a 50-hectare mapped plot. So it's 1,000 meters by 500 meters, and every single stem in that plot is mapped. And so each of the dots on this, in each of these rectangles, is an individual of that species. So we have here 2 Dryabellinop species, a Shoria and a Hopia, and then Swintonia here. And what we see, so one of the factors that can really contribute to that kind of clumped distribution can be habitat availability. So here we have our habitat classification. So we have 3 different habitats. if I'm remembering correctly from the paper, this is sort of a ridge structure here, so higher up, drier, sandy soils, and this is kind of a low-lying alluvial area. But it could be flipped. But that isn't really the point I'm trying to make here. The point is that if we take a look at the distribution of some of these different species, we see very highly clumped distributions. So Hopia dryabellinoides, for example, only occurs where we have this light gray habitat type. Okay, so kind of in this hilltop environment. In contrast, Dryabellinops aromatica only occurs in this lower lying alluvial habitat down here, this floodplain habitat. So we have these two contrasting distributions, which we refer to as habitat specialization, but a very strongly clumped distribution that goes hand in hand with that abiotic environment. There are other factors that can lead to that sort of clumping as well, but this is just a really dramatic example of that. And then you have other species like Schorialaxa that tend to have a much more generalist distribution across these habitat types. There still seems to be some clumping, and that can be because of dispersal factors. You can imagine that these big seeds don't drop very far from the parent tree, even though they are wind dispersed. You can see wings, and so there are dispersal related reasons that we see clumping as well. Okay, so clumped distributions in tropical rainforest trees. Another important thing to think about when we're talking about, population distributions and abundance is the geographic range size of the species. So when we're talking about the geographic range, this is the entire geographic region over which that species is found. And this includes areas that it occupies at all life stages. This is an important piece because, when you can think of migratory species. And, our known experience with those species is just the habitats that they occupy while they're, physically present in our space. But those individuals move around a lot. And the most extreme example of that is here with the Arctic tern, where it spends its summers, its nesting season, so it nests in the Arctic. And you can see up here various parts of Western Europe and Scandinavia. Then it migrates down to Antarctica for the Antarctic summer or the Arctic winter. So it spends the Arctic summer up at high latitudes. It spends the Antarctic summer or the Arctic winter down at very, very low latitudes, and then migrates between them, between those two seasons. So when you're thinking about geographic range size, you really have to be capturing all of those different components of a species' life history. And sometimes it's much less extreme than this, of course. Most times you can think of something like a frog that maybe spends, it requires aquatic environments for juvenile stages, but then spends its adult stages predominantly in forested environments. So when we think about the geographic range, again, that encompasses all of those different habitat types. And when we think about geographic ranges, there are different types of distributions. And these are the two kind of extremes here, but on the very sort of most limited and smallest extent is an endemic species. So a taxa that is native to a certain limited area. They often receive sort of special importance because they're found only in one location and nowhere else on the planet. So when you hear the word endemic, it refers to a species that is limited in its extent to that particular geographic region. And so if that species goes extinct, it's not found anywhere else on the planet. And so when we're thinking about conservation initiatives, those components of the geographic range are taken into consideration in important ways. How important is this population for maintaining this species? So, a great example of this is this Montezuma well in Arizona. There are several species of, at least five endemic fish species that occur just in this one single sinkhole. And so, That's a very extreme example of endemism. A lot of times the species is more widespread than that, but restricted to a single location. But this is a really extreme example. On the other hand, we have cosmopolitan species whose ranges extend across all or most of the world in anywhere where the habitat is appropriate. And an example of this, and we're going to see this friend later in the lecture as well. An example of this is killer whales, whose distribution basically is depicted here by blue. So anywhere pretty much on the planet, killer whales can be found in ocean environments, of course. Many species do not have distributions that are contiguous across an entire area, and in fact show very patchy distributions. And so here we have this example of the red kangaroo in Australia, and we have a map of its distributions, and these different colors correspond to different densities. So over here in southeastern Australia, we have very high population densities of this particular species. And then this broader range that extends over to the western coast of Australia, where populations are pretty low throughout much of it. We have some areas where they're getting a little bit higher. A lot of this has to do with habitat suitability through the range. Then you can also see all of these gray areas where red kangaroo just simply does not occur. And so the habitat conditions there, either the habitat conditions or some aspect of the biotic environment are meaning that species cannot occur in those areas. So much of coastal Australia, red kangaroo is absent from. And in these really interior areas, we see these gray patches as well where it's absent. Okay, so we can have naturally patchy distributions. We can also have, increasingly in the world today, we have patchy distributions that are caused by human disturbance to habitats, human modification of the landscape. And this is a great example of this. This map shows the distribution of these beautiful heathlands in the Dorset region. So here we have Great Britain. We're looking at this little subset in Dorset. And the yellow areas reflect the distribution of these beautiful heathlands. So heathlands tend to be dominated by ericaceous shrubs. So you can think, if a favorite ericoid is blueberries for many people. So ericaceous species dominate these heathlands. And you can see that in 1759, the extent of those heathlands was really broad and very contiguous. There was some patchiness in the landscape, but pretty large patches and pretty close together. By 1978, those patches had been reduced greatly because of urbanization and farming. So by 1978, those patches were restricted in the landscape to fairly small and isolated patches. And so this is going to be a focus of a lot of our discussion today, is these patches, movement of individuals amongst those patches, and sometimes individuals can move between patches, but this depends on what is in between. So in a naturally fragmented landscape, it may be the case that whatever is in between the patches is amenable to moving through. Very oftentimes in human modified landscapes, this isn't always going to be the case. And we refer to that area between patches as the matrix. So between any of these red patches, or in 1759, this green area between the yellow patches would have been the matrix. And in 1978, now this yellow area and the green area has become the matrix. And so depending on what's there, this ranges from, paved urban areas to, some other type of open landscape that maybe is all right for individuals to move through. So the quality of that matrix dictates how common and how difficult dispersal will be between patches. Okay, and so sometimes we also don't know the true distribution of a species. And so what we'll often do to deal with that problem is that we'll characterize how biotic and abiotic factors impact the occurrence or abundance of a species. And so the way we do that is if we know where that species does occur, we quantify its abundance or its occurrence, depending on what you're interested in, and we quantify the environmental and the biotic, so the biotic and abiotic conditions at that location. And then we can use that information. So if you think about, carving the landscape up into little grid cells where you say, okay, is that species present or absent? And what are the environmental conditions and what are some of the other biotic conditions that may be important? Then we can model this, especially when we're talking about environmental factors. It's harder to model the biotic components. But then we can model this and make predictions of where we expect that species to be on the landscape. And these are referred to as species distribution models. So we relate these habitat characteristics that we can map on the landscape to the known requirements from a habitat perspective of a species. And so this can not only allow us to come up with a prediction of where that species should be in the landscape, but it can allow us to identify places where maybe we don't know that species is present, and we can go in and actually identify whether it's there. And from a conservation standpoint, this is really valuable because we can find new populations in some cases and identify places that will be really important for protection, even if they aren't occupied. So an example of this from your text has to do with chameleons in Madagascar, these lovely lizards. Okay, so Madagascar being down here off of the southeastern coast of Africa, and what researchers did, they were really interested in chameleons, and there were, I believe they modeled about 11 different species of chameleons. And what they did was exactly what we just talked about. For all the species they were interested in, they knew their current abundance or distribution, so their current occurrence or abundance, and they knew the environmental conditions that those locations were characterized by. And by doing that, they were able to map use species distribution modeling techniques to map the anticipated distribution of each of these species. And what these circles indicate on the map are places that the species distribution models predicted should have that particular species of chameleon, so in this case the panther chameleon, but where it was not known to occur prior to that effort. And so they were actually able to go out into the environment and identify places where these populations did occur that they didn't know about before. Okay, so this can be a really incredibly valuable tool when we're thinking about conservation planning and range management planning for different species. Okay, and so for each of these, the spiny chameleon, the plated leaf chameleon, these circles represent the same thing. Lots of new areas were identified for each of these species where the species was not known to occur previously. Okay, and in addition to that, while they were out doing these surveys of this new territory for these different species, they identified additional species within those areas where there was overlap in so we have overlap in these circles. A lot of additional species also use those areas. So this is a really incredible tool for thinking about for applying our knowledge of the distribution of species and their environments that they occupy to produce expected maps of the expected distribution, which can then allow for new discovery. Okay, so we know that habitat suitability is going to determine distribution and abundance. So we can go back to this, this figure that we've looked at a number of times, which gives us information about the abundance of an organism across some environmental gradient. So we know in order for a species to be present in a particular location, the environmental conditions have to match its its requirements. So we need this suitable habitat, okay, and this gives us this potential distribution. And this potential distribution, as you are all aware, is determined largely by these abiotic factors. And so we can take a look at this in the context of these two desert species, the creosote bush. We're revisiting this. We saw this in the first lecture this week. The creosote bush and the saguaro cactus. And these are two species that co-occur in deserts in California and Arizona and right down into Mexico. And we have here the range of each of these two species. So the red range corresponds to the range of the creosote bush, which you can see is much, much broader than the more limited distribution of the saguaro cactus, which is here in the brown color. Okay, so the creosote bush extends both further south and further north than the saguaro cactus. Okay, so both of these species, this is a very arid environment, both of these species have a tremendous tolerance of arid conditions, and they do this using using different physiological mechanisms. You can see these tremendous difference in the morphology of these species. They use different physiological mechanisms to deal with drought, but a key difference that drives this differential distribution is the fact that the saguaro cactus is not able to tolerate freezing. So its northern limits are restricted to those areas where freezing temperatures are not maintained for more than 36 hours. And so it just is unable to deal with cold temperatures that you can get as you move further inland or further north in these desert environments. Okay, so this is the kind of information we can get relating our abiotic conditions, or to using abiotic factors to understand the drivers of distributions of different species. There's also biotic factors, and if we go back to this figure, if you recall, those biotic factors are what limit from within this potential distribution that's driven by abiotic conditions. These biotic factors are what restrict that niche, what lead to the realized niche as opposed to the fundamental niche. And so it's those biotic factors that lead to this actual distribution of a species. And so in this case, we're looking at this lovely little warbler, Seychelles warbler. Here's the Seychelles Islands. Here's Madagascar. We just visited Madagascar talking about those chameleons, and we're up here in this little set of islands. And this Seychelles warbler is an endangered species. It occurs only on this single island, cousin island. And these are highly territorial birds. They form mating pairs and defend their territories. And so you can see this whole region carved up into territories. Now, what you also see in this map is territory quality, and this has to do with the availability of food resources. These warblers feed on insects, and so it really has to do with the availability of those preferred insect species on which they feed. And so in these darker yellow areas, we have a high mean territory quality. In the lighter brown areas, it's a low mean territory quality. Now, this drives differences in abundance of the Seychelles warbler in this habitat, because what will happen is in the areas where you have high territory quality, juveniles from previous years will actually stick around and help care for the young of year, because there's sufficient food resources to support those recently fledged individuals. So you have larger population density. So it's not just the mating pair and their young of year that are in each of these territories that are dark yellow, but they can also maintain their young from previous years who then help to help with caring for the nest, bringing food to the nest, all of these sorts of things. And so the survivorship of the nestlings is higher in these areas with higher habitat quality compared to these areas with lower habitat quality, where there's not sufficient food resources to support that kind of family unit, if you will. And so we have this difference in territory quality that drives strong differences and clustering in the population abundance within this species on this island. So a great example of how biotic resources can dictate what we're seeing in terms of, in this case, in terms of abundance, in terms of the numbers of individuals in a particular area. So we have much higher abundance in this dark yellow area compared to the lighter yellow compared to the brown. We can also think of other biotic factors. So here's a picture of what we refer to as a herbivore exclosure. This is simply a fence, and in this case it's keeping ungulates out, it's keeping deer out, who when they're in high densities can lead to this kind of situation where basically it looks like nothing is recruiting in the understory. But when you exclude them from the picture, all of a sudden we have all these different species that are growing up in this area. So it just demonstrates that the environment is a match for those species, but the biotic pressures are meaning that those species are not able to occur where the biotic pressure is present. Okay, so another example of those biotic factors that can limit or can lead to a patchy distribution. Okay, and then here's a nice example of where we actually see these joint effects leading to a difference in geographic range, okay? So here we have this barnacle that we've seen before, S. balanoides, and based on its thermal tolerances, it does very well in these cold environments fairly far north. So here we have the Aleutian Islands, Alaska, we're heading down there's Vancouver Island, heading down into British Columbia here on the west coast. So we have these colder ocean waters here through the Bering Strait, and down here, the temperatures in the waters down here are fine from a physiological perspective for S. balanoides, but we actually don't see it present in those areas. And the hypothesized factor that drives that is that competition with other barnacles prevents this species of barnacle from living here, despite the fact that the temperatures of the water and that the ocean conditions are suitable for its presence in that region. So its range is restricted to these colder areas where some of those other competing barnacle species are not able to occur. All right, so this is a great example where that potential or the fundamental niche is reduced, potential distribution or fundamental niche is reduced to a smaller actual distribution or realized niche based on biotic pressures. Okay, disturbance is also a factor that is really critical for species distributions and for species distributions through space and time. Okay, so a disturbance is an abiotic event. So this is critical. An abiotic event, you can think of flooding, you can think of fire, you can think of different types of storms. An abiotic event that kills or damages some individuals and thereby creates opportunities for other individuals to grow and reproduce. Okay, so here's a great example. This is from the Pacific Northwest, so not too far from those coastal S. balanoides populations, the forest just inland from them, and we have here a map of the vertical and horizontal profile through a 140-year-old Douglas fir stand. So all of these PMs are Pseudotsuba menziesii or Douglas fir, and then we have different species that are here in the understory, kind of just sitting there, not doing really all that much, not doing necessarily very well. But their maintenance in this system is really driven very critically by the fact that as these trees get older and taller, eventually wind will, wind and or other disturbances will cause them to snap off in some cases. In some cases it's just senescence, but in some cases we end up with storm events that open up these canopy, the forest canopy, allowing additional resources, specifically light resources. These tall temperate rainforests are really light limited, allowing light resources into the understory, and supporting the growth and the growth and productivity of these other species that occur in the understory of this forest. So here you see the map of where the canopies are overlapping in this 140-year-old Douglas fir stand. So this is pretty young. Those Douglas fir have grown up, and they're just shading out all of these species in the understory. You can see the overlap of their crowns depicted by these circles in the map, okay? Then at 450 years, we've got all of, those trees have gotten to a point where they're no longer structurally sound, and factors like wind and storms and different abiotic factors cause them to snap off, and we end up with just a couple of adult individuals remaining in the canopy. So we see their canopies depicted here, and then this understory, the understory vegetation growing up, and that will form the next canopy. Okay, so we have these processes, these disturbance processes, that are really important for maintaining forest diversity, for example, and maintaining different species within these environments. You can think of processes like fire in this context as well, where you have a fire that burns through the system and leads to secondary succession. So this transition from through this sequence of vegetation that eventually leads to a mature stand, in this case a mature oak and hickory forest. But in the interim supports a lot of different species that would not do very well in this mature forest, but do very well in these different successional stages that occur following the disturbance. Another critical thing to keep in mind when you're thinking about species distributions is that the way the world is configured and looks today is not the way it has always been. So here we have the configuration of the different continents through geologic time. So here we have Pangea, where all of the continents were in a single supercontinent, and the breaking up of Pangea to something akin to what we have today. It's a little bit of a rough diagram of that. But this gives you some inklings into changes where different land masses were connected in the past. This can give us insights into why the distributions of certain organisms are the way they are, despite the fact that maybe those land masses are separated now. So for example, we have South America and Africa connected here during the Jurassic. They are now separated by a very large ocean, but there will be some shared biodiversity components because of the fact that they were connected through a long period of evolutionary, of geologic time. Okay, and this map shows something similar but a little bit different. It shows how the distribution of biomes has changed as those plates. So here again we have a supercontinent, and as those plates moved around and gotten to something closer to what their present day configuration is, we see how those different biomes have changed through time. And again, this history of these sites can be a really important clue into why certain species are where they are, why there are certain relict species present in different places, etc. And so thinking about this much longer term geologic history of the planet and the associated evolutionary history that goes along with that. And a great example of this comes from this region over here in Southeast Asia and Australia. we have these regions that are pretty close together, and a whole series of islands that kind of span from over here we have Indonesia and Malaysia, Borneo. We were just looking at those tropical rainforests, the dipterocarp forests here in Borneo. Then we have these islands that kind of connect over here to Papua New Guinea, Australia. But so Alfred Russel Wallace, who is, who's considered the father of biogeography, this was a really key discovery in his scientific career, was that the fauna of these two regions was really quite distinct. The most notable difference is that we, to the southeast of these lines, and so here we have Wallace's line, which he drew in 1859, and this was revised by Weber in the 1900s. But here we have, down here to the southeast of the line, we have a lot of marsupials and monotremes, so these really unusual mammals that you find in Australia and New Guinea and New Zealand. And then to the northwest of the line, we have placental mammals, like we have in many parts of the world. And so this was a big surprise, because a lot of these islands, it looks like you could, there are, potential, there in the past have been land bridges between different parts of this when sea levels have risen and fallen. And so the question for Wallace and for Weber was, well, why are these fauna so different in these two regions? And we can look to this map that we have of the configuration of the continents through time, and this gives us some insights. So, for example, here we see that Australia in the Jurassic and the Cretaceous was connected to Antarctica, right? It was not connected to the places, so now in present day it's very close to Southeast Asia and to this whole kind of archipelago region, but through most of geologic history it was not. And so here we have Southeast Asia and Australia pretty close to one another now, but at that point in time that was not the case, and so they were very separate from one another for very, very long periods of time, which led to different evolutionary trajectories for these different land masses. And then through plate tectonics, they came close together, but those evolutionary histories driven by the geologic history of the planet and this separation, the total vicariance of these areas, is what drives these differences that we see in the fauna that we have in present day. Okay, so dispersal and migration. Oh, I don't know why there's all this animation on here. Sorry about that. So a couple of definitions. Dispersal is the movement of organisms away from their point of origin. Okay, and so here we have this dandelion with its wind-dispersed seeds, and those wind-dispersed seeds are being moved away from the adult plant. In contrast, migration is the movement of individuals and commonly whole populations from one region to another. And here we have the case of these whales that spend their winters down here. This is their winter breeding area down here around Hawaii. In these kind of tropical areas, they spend their winter in various breeding locations. And then they all move northward in the summer to feed. When the cold oceans are productive, they move northward and feed in those areas, and then they return south in the winter to breed again. Okay, so this seasonal movement of populations, individuals, and typically entire populations from one region to another and back. This is what we refer to as migration. And when we think about the habitat requirements, when we think about the geographic range of a species like this, it's all of those areas that it's using, okay? All right. Okay, and I just want to touch very briefly on the evolutionary role of dispersal. This is really important. Dispersal is important for organisms for a number of reasons. So, when we're thinking about individuals, what does dispersal do? Well, it leads to the discovery of new habitats. If you are an individual living in location A, your offspring are dispersing away from you, and maybe they find a new habitat that is better than what you're in. Again, the whole goal of reproduction is passing on your genes to the next generation, and ideally those offspring are able to pass on more genes to the next generation. So by dispersing organisms away from yourself, those new individuals may find new habitats. That those new habitats may well be better than the current habitat. It could also be worse. So there's a bit of a gamble there. For some species, dispersal may be necessary to complete a life cycle, and this is true for many parasites, for example. We think about the life cycles of parasites where they have to move from one host to another and maybe into the environment and back into a host. And so dispersal in that case is necessary to complete the life cycle. And when we're thinking about dispersal, the important things to consider is habitat similarity and quality. So if you have a lot of, dispersal is most advantageous if the habitat that the individual is being dispersed to is a suitable habitat for that individual, a high quality habitat, so similar to the habitat that its parent is on, and or a better habitat. And so what we often see is that organisms will disperse. The most common outcome is that organisms will disperse some distance away from the adult, that the frequency of organisms dispersing away from their adult, from their parent, is high, but that the most frequent outcome is dispersal, that a dispersal distance that's just far enough away from the adult. So it moves that individual away from its parent, but not so far away that you're taking a big gamble in terms of what the new environment is going to look like. Okay, so yes, here we have spreading away from a birthplace. So we have pros and cons. So the advantage is reduces the competition with the parent. So if you think about a tree that produces seeds, those seeds that disperse away from the parent won't be shaded by that parent. You know that if you're the same species, that you have the same resource requirements. So if you're growing too close to your parent, you're going to be competing with your parent for those resources. So that can be a disadvantage. Dispersing away from the parent also reduces competition with siblings. So again, if we think about the distribution of seeds from a tree, many of them fall right below the tree. And if you're able to disperse away from your parent, this gives you an advantage over your siblings for a number of reasons. One is competition for resources, but also there's aspects of pathogens and predators that if you have a high density of seeds and or seedlings around a parent plant, that you're more likely to die because of infections or because of seed predation or herbivory. Okay, and this is referred to as the Jansen-Connell effect. Okay, and another advantage, I mentioned this already, there's a chance that you find a similar or possibly better environment than your parent has found, okay? So, the disadvantage, of course, is that you disperse and you land in a place that is terrible and you can't grow and you don't pass your genes on to the next generation. So, there's pros and cons, and this is where this trade-off comes in, that natural selection tends to favor individuals that move some distance away from the parent, but not too far away from the parent, okay? So this is the general observation in terms of frequency of these dispersal distance outcomes. All right, so dispersal is really important, moving from away from a parent, but also dispersal is really important in the context of population dynamics. So there's what we refer to as metapopulations, and we've already seen this definition, a population of populations. And so here is a picture of this. We have 1, 2, 3, 4, 5, 6, 7 different patches that are suitable for a particular species to occur in. All of these areas have the potential to support populations. In a metapopulation, we typically think about kind of this blinking on and off of these different patches, that some patches will support populations, other patches will have suitable habitat to support populations, but will not have individuals present. And so movement of individuals from one patch to another can lead to colonization of suitable habitat that's not already colonized, that's not already occupied, or it can support the maintenance of populations that are occupied by moving individuals from one area to another. So this is what we refer to as metapopulation dynamics, or a population of populations. And so it's this set of spatially isolated populations linked to one another by dispersal. So dispersal is key in all of this. Okay, there are two pieces of this, and we'll look at what source and sink populations look like. But when we think about metapopulations, there are sources where the number of individuals that disperse is larger than the number of individuals that arrive. This can be attributed to a number of factors. A key factor is often either the size of the patch or the quality of the patch. That leads to higher reproductive rates and the ability for more individuals to emigrate from that population. Okay, so high birth rates in a particular patch can lead to greater emigration to find new habitats. Again, if you have a single patch and your birth rates are high, eventually that patch is going to get crowded, and it's going to be in your best interest to try to find someplace where you can find a new patch where population densities are not so high. So these are source populations. Sinks are populations that receive more immigrants than the number of emigrants that they produce, okay? So this will tend to be populations that are occurring in habitats that are lower quality, where death rates are higher, birth rates are lower, or both. And so as a consequence, they export fewer individuals from that population, and a lot of times that population may be maintained by individuals coming in from a source population. Okay? So again, it's this idea of a blinking on and off of populations, and so we can think about it in this context. And there's all kinds of different types of metapopulations. And we see them here from kind of a situation of very high connectivity, where you have a continuous habitat, to patchy but very well connected, so some fragmented habitat where there's a lot of movement amongst individuals in the population, and all of those patches are occupied all of the time, to mainland island core satellite. So you have a mainland and a bunch of islands, or a core population and a bunch of satellite populations. And the dynamics here often have to do with the distance of those core, the satellite or island populations from this core or main population. So you can see that populations that are closer to this main population are being occupied, whereas populations that are further away are not. This is referred to as the theory of island biogeography. And if you take the biogeography course, the biogeography and conservation course, you will learn all about this. Then here we have a metapopulation. Okay, so these are not metapopulations. These are different configurations of populations related to connectivity. Here we have a metapopulation where we actually have, individuals moving from one population to another, and we have information about newly colonized populations. So these ones with dashed lines around the outside of them, these are newly colonized. Individuals have moved from one population to an uncolonized patch, and it's newly colonized. We also have populations that blink off. So this is the blinking on, this is the blinking off. These are patches where local extinction has recently happened, and that population blinks off. Okay, so this is our idea of metapopulation compared to these other types of population dynamics. And then here we have what we refer to as a non-equilibrium population, where we don't have movement of individuals amongst patches for various reasons, and the majority of populations have gone locally extinct. And so that a population like this or a population of populations like this is sort of on its way out the door. So all of these are populations of populations, but when we think about metapopulation dynamics, it's this blinking on and off of, these new colonizations and extinctions that drive dynamics in the landscape in terms of where individuals are in the landscape. And the health of that metapopulation is really dictated as it's the balance between extinction and colonization. And so Richard Levins was the first one to sort of to identify these dynamics and to put together an actual model to model those dynamics. And so he was the first to represent metapopulation dynamics. And it's a fairly straightforward equation. So the change in your population, or the change in the proportion of patches occupied at time t over the change in time. So here we have a rate, is a function of colonization of empty patches. So C is our colonization rate, P is the proportion of patches occupied at time t, and this is our proportion of patches not occupied at time t. So here we have our colonization function of the model. Here we have our extinction function of the model. So our rate of extinction times the proportion of patches occupied at time t. Okay? So two components in this model. One focusing on the colonization piece. So the colonization rate is a function of how many patches are colonized. What is the rate of colonization? How much do those individuals move out from those colonized patches to uncolonized patches, and how much of the landscape is uncolonized? And then extinction is just acting directly on that proportion of patches that is colonized at time t. Okay, so there are some assumptions, and we've already seen that these assumptions probably aren't always holding true in real life. In fact, frequently don't. So there's a very large number of identical habitat patches. So we've already talked about source sink dynamics. So we know that in most cases, habitat patches will not be identical. Okay? All patches have an equal chance of receiving colonists, so the spatial arrangement doesn't matter. Again, and I'll show you some data on this, but we know this is probably not the case. If you think about an individual that's trying to disperse to, a patch that's right next door to it versus a patch that's a kilometer away, it's a much higher likelihood that you're going to move to that neighboring patch as opposed to the more distant patch, okay? So spatial arrangement doesn't matter, and all patches have an equal chance of extinction. And again, when we think about those ideas of source sink that we just talked about, or what we know about habitats in the landscape, what we just, for example, what we just heard about the Seychelles warbler example, where of course different patches of habitat are not equal. This often is not the case. So in many cases, our patches do not have an equal chance of extinction. But these are the assumptions that go into this particular model. And this model has been incredibly useful for understanding the dynamics of populations in the landscapes, and for predicting or anticipating the impacts of habitat fragmentation driven by humans on populations of different species. So this has been an invaluable step forward in our understanding of how a fragmented landscape functions, okay? But the take-home message from all of this, from this particular equation, is that for metapopulation to exist for a long time, so for that population of populations to exist through time, your ratio of extinction to colonization has to be less than 1. So your colonization rates have to be higher than your extinction rates, okay? All right, so the important links of this metapopulation theory have to do with habitat fragmentation. And so we see this increasingly fragmented landscape here with decreasing connectivity and growing distances between patches and decreasing patch sizes. All of these things have the real potential to decrease the likelihood that metapopulation is going to persist, because what this kind of fragmentation of a landscape does, it can reduce colonization rates. Of course, it's really hard to move across this open landscape. If you're a forest species, you're trying to recolonize this patch over here. For many species, moving across this landscape is going to be really hard. Okay, so your colonization rates decrease as the landscape becomes more fragmented and patch sizes become smaller. It's also harder for a patch to support a population that can export individuals if the patch size is smaller. So fragmentation leads to greater barriers to dispersal. This can reduce colonization rates. Extinction rates may also increase as fragments become smaller and more isolated. We know that a smaller fragment is that the likelihood of extinction increases as your patch size decreases. So the smaller the fragment you have, the more likely you are to lose those individuals, simply because you have a smaller population there, and different events can act to wipe out those small populations. Okay. So with sufficient fragmentation, what happens is extinction rates exceed colonization rates, and we now move into a situation where that E/C ratio is less than 1, and that metapopulation is kind of doomed to extinction. So habitat fragmentation, which we see in such a widespread way throughout the world, has a really big impact on the likelihood of these metapopulations to be maintained. Okay, and here's this aspect of isolation by distance. So if you recall, one of Levin's assumptions in his model was that, sorry, I have a little scratchy throat today. One of his assumptions was that all patches were equally likely to be colonized. And here we have information on, or a graph showing information on patches that have suitable habitat for this particular skipper butterfly, Hesperia coma. So this species is found in calcareous grasslands, and so these individuals, the authors of this paper, went out and mapped all of the appropriate habitat for that particular butterfly species. In the early 1900s, the habitat available for this species was really widespread because of agricultural uses. So these calcareous grasslands were really common at that time. Agricultural intensity decreased in the 1950s, so lack of grazing cattle was reduced on the landscape, and so these grasslands became overgrown, and the habitat quality declined for this species. And then starting around the 1970s, by the 1970s, there were only about 10 restricted grasslands. And so the population of the species really declined quite dramatically. Livestock numbers in the 1980s started to rebound in the landscape, and so more of this grassland became available. So these individuals, the authors of this paper, went out and mapped the available habitats and looked at both patch area, so how large was the fragment on the landscape, and how distant was it from the nearest occupied patch, okay? And then they looked at whether or not in 1991 that patch was colonized or not colonized, okay? And what we see here, these lines represent different probabilities of colonization. So anything that's here or beyond has a 90% or greater chance of being colonized. From here, in this area, we've got about a 50% chance of being colonized, and here only a 10% chance of being colonized. So you can see that places in the landscape that had large patches that were close, so these are small distances from nearest occupied patch, so large patches close to the nearest occupied patch had a very high probability of being occupied. Small patches that were distant from other occupied patches had a very low probability of being occupied, okay? And so this demonstrated the real importance of isolation of these fragments in the landscape, both isolation and patch size, in determining the colonization outcomes. Okay, and so this speaks to the importance of what we refer to as the rescue effect. So as you will have come to realize through this lecture, despite those assumptions of Levin's model, not all patches are equally good, and not all populations have equal chances of extinction. So we have some patches that are better quality, either because they're larger or because the habitat quality is better. This leads to differences in the likelihood of extinction. But there's also this component of the rescue effect. even if a population does go extinct, if you're close enough to a population where that is producing emigrants, then you may be rescued from extinction. Okay, so high rates of immigration from a neighboring source area, so a higher quality area, can help to prevent extinction where a patch is small or lower quality. And we know that likelihood of colonization is going to be greater if you're closer to an occupied patch. So we can think about this context where we have some habitats that are much better. So in this case, we have much larger habitat areas. Maybe they're also higher quality. We refer to these as source populations. And source populations send out individuals into the neighboring sink populations, okay? And so they, in many cases, they rescue these sinks from going extinct. It may be the case that these smaller patches are too small to support a viable population. It may be the case that the habitat quality in those populations is not great and does not support a viable population. Either way, when you have a neighboring population in a metapopulation, a neighboring population that has high rates of emigrants that can move into these neighboring sink patches, it leads to what we refer to as the rescue effect, or the avoidance of going extinct because of high rates of immigration. Okay, so we are almost to the end. We're back to the mystery of the kelp forests. Okay, so if you remember, we had these kelp forests that were declining. So the distribution of these important dominant species in these coastal marine ecosystems, they were declining, and this was having big impacts on the health of those coastal areas. And in fact, here's some information. This is not from the same area that's in your text. This is from the coast of California. But we can see that in 2008 there were kelp forests all along the coast here, and so we see the distribution here depicted in these green dots. By 2019 there were no kelp forests remaining. So this was fairly alarming to researchers. And in many cases this was, or in all cases, this was what it looked like. Moving from these gorgeous kelp forests that support all of this life to these kind of desolate barren lands that are covered in these sea urchins. And if you recall from our first couple of slides, the removal of sea urchins helped to promote the recovery of kelp. So we know that these are the culprits in this mystery of the disappearing kelp. But this is what these places look like. You move from this lush, productive environment to this kind of moonscape where everything is decimated and it no longer supports that level of biodiversity or productivity. So it's a fairly complicated story that involves a lot of different factors. And I thought it was really important to highlight the importance of some of these different components of species ranges that we've talked about through this seminar, and the importance of some of the different climate change and anthropogenic pressures that we're putting on populations that can really drive quite profound changes. So one piece, and this is not highlighted in your text, but I'll pop the link to this NPR article that I took that map from, and I took these images from. One important piece is that climate warming is driving the loss of this really key predator of sea urchins. So this is the sunflower star. Is a very large starfish, and you can see that it actually here it has an urchin. It's enveloped an urchin in its arms, and it's consuming that urchin. Okay, so this is a key predator of urchins in this ecosystem. Climate warming is driving warming ocean temperatures, which make these sea stars more susceptible to various diseases. And most notably, between that 2008 and 2019 map that we saw, there was a really large warming event in the Pacific Ocean called the blob. It was very, very hot, warm ocean temperatures that drove declines in a lot of different species, including these sea stars. And this continued warming is leading to increased disease, and now this is in this portion of California an endangered species. So we lose a really critical predator. a second predator was also declined substantially for a couple of key reasons. One is these lovely little otters were, they feed really voraciously on these sea urchins. These lovely otters were, in the 1800s, hunted pretty heavily for their pelts. So the fur trade led to really rapid declines in the populations of these, and that led to proliferation of sea urchins. But more recently, there's an additional pressure in some of these large predators in the ocean, so namely the killer whale, which we saw earlier as a cosmopolitan species. Killer whales prefer to feed on really large. They hunt in packs, and they feed on larger whales. Those larger whales have been overexploited. So just like the sea otter was overexploited in the 1800s, these larger whales, which serve as a typical prey species for killer whale, have been overexploited, and their numbers have declined dramatically in the oceans. This has led the killer whale to look for different food resources, including various types of smaller mammals like seals and otters. And so greater predation by killer whales has also contributed to more recent declines in sea otter populations. And so we've seen that here from 1985 to 1997, this pretty dramatic decline in the abundance of otters in a variety of places, from the Aleutian Islands, which we saw earlier, up into Alaska. So a different part of the coast. We're not in California any longer, but same kinds of ideas, same kinds of problems. So without these declines, so we saw without this additional pressure on the sea otters are able to consume a lot of sea urchins, and the sea urchins have put much less pressure on the kelp. When we have reduced populations of sea otters, whether due to harvesting of the otters themselves or predation on the otters by killer whales, this can lead to a reduced pressure on the sea urchins, which then have a higher than normal population and consume and lead to the removal of kelp. So we see this relationship that when sea otter populations were high, urchin biomass was low, and urchin grazing intensity was low. As those otter populations decline, we see this increase in urchin biomass. Here on the y-axis, we see this increase in grazing intensity, and then we see the opposite happening for kelp. So when otter populations are high, we see a high density of kelp in the oceans. When otter populations are low and urchin populations are high, we see this decline in kelp abundance. And so we have these various factors at play that are driving the reduction in predators on sea urchins, and so then the sea urchin populations are able to explode and decimate these kelp regions. And there's an interesting source-sink kind of dynamic at play in places where there has been reintroduction of otters. We have two regions, one in southern Alaska, one in the Aleutian Islands, and you see very different responses. And so what we're seeing here, the blue dot is where otters are absent, the black dot is where otters are present, okay? And so in southern Alaska, anywhere where there's otters present, urchin biomass decreases dramatically and kelp density increases. So we have kelp density on the y-axis, urchin biomass on the x-axis. The blue dots are otters absent, so where otters are absent, consistently we have higher urchin abundance and very low kelp abundance. Where otters are present, the black dot, we have low urchin abundance, high kelp abundance. So we have this clear relationship in southern Alaska. And there's some indication of that in the Aleutian Islands as well, but it's really not as clear. So here we have, again, the blue dots are where otters are absent, and in some cases there's really no change between the otters absence and otters present populations in terms of either the urchin biomass or the kelp density. And in some cases the urchin biomass decreases, but the kelp density doesn't really recover. And so this was a little bit of a puzzle, and what researchers found was that in fact these areas in the Aleutian Islands are being recolonized really regularly by, so the sea urchins have a planktonic larval form, and this is being carried by ocean currents to these Aleutian areas. And so despite the fact that urchin populations were being knocked back, by the otters, recolonization was happening pretty readily from other places, bringing individuals, bringing new recruits into the population, new individuals into the population, and maintaining the pressure on the kelp from the sea urchins, which led to a slower or non-existent recovery. So all kinds of really interesting different dynamics at play in this particular story, but a lot of complexity where we have, climate change acting as a pressure, changes in biotic interactions via predator-prey dynamics at multiple levels in this food web, and changes in population dynamics of the urchin population due to dispersal into these populations where otters are present. So a very complicated story that took a lot of work to unravel, and so there's a lot of effort going on to sort of restore some of these kelp forests and figure out ways to help those areas recover. So with that, that's the end of the lecture for today, and I will see you this evening. Have a great day.